So welcome everybody to the autumn seminars from the EVOLTREE network. Good to see so many of you here. It's great to be back. It's been a year since we ran the last series, and we had lots of good engagement last year, so we can have the same again this year. Just before we get started, I just want to remind everybody briefly that this set of seminars is coming from the EVOLTREE network, which is an EU network which has been running now for over 15 years, I think. And there's lots of information on the website that we have here, www.evoltree.eu. So if you do want to follow up on anything, then please go there. I'm just going to advertise a few things that we do offer, though, particularly a couple of things coming up which are current. So the network offers some small grants through the opportunity call, and we have a call open at the moment, so that's open to all researchers associated with the EVOLTREE network, but is got a closing date of the 5th of January. It's a very straightforward application, but do have a look if you have an idea. We particularly welcome applications from early career researchers, but it's not limited to those. We've got a series of training courses as well. There'll be a training program that will open and run through all of 2025. There are a couple of courses on there that are open right now. Do have a look at those, and the instructions for signing up or applying are available there. The network can support a few travel grants to training courses, so if you need support to be able to attend, then do have a look at that. But again, a very simple application process. And I guess the main thing I'd like you all to hold the date for next year is the EVOLTREE conference, which will be happening in Madrid, 17th to 21st of November. I think that should be. It's not a 2-week conference. We had a great meeting last year, 2023, in Romania. It was really good to get everybody back together again in person. I think that was the first big event we've had since the pandemic. Lots of good attendance, lots of interesting talks, and a great chance to spend some dedicated time hearing about forest genetics. So please hold that date and look out for future announcements. So I just want to, before we introduce today's speakers and this series, just to flag up that the previous seminar series that EVOLTREE have run have all been recorded and made available on the YouTube channel. There's lots of good talks there, so please do go and have a look. We've had 5 different series now, so there's lots of content, and in fact all the talks from the previous EVOLTREE conference are now uploaded there too, so there really is a lot to look at. So please follow up on that if you've got anything you'd like to hear. And then we're going to be presenting 2 seminars this autumn. In 2 weeks' time, I hope you'll come back for a future-oriented deep time perspective on European woodlands, which will be given by Professor Jens Christian Spenning from Aarhus University, and that promises to be very interesting, so do come back. But today we're going to be hearing about hidden diversity and dynamics in rainforest trees, so moving right away from Europe, and we'll be hearing from speakers today, Professor Catherine Kidner and Dr. Rowan Schley. Catherine is at the University of Edinburgh and the Royal Botanic Garden Edinburgh, and Rowan is a postdoc working with her, but here and at the University of Exeter. So without further ado, I'm going to hand over to Catherine to kick us off.
Thanks very much. I'll share the slides. I'll take them up right. Everyone can see that clearly, yeah?
Yeah, that's fine.
Okay. I'm just going to make everyone's pictures disappear so that we can see the whole slides whilst we're talking. So shout out if you have questions, because I won't be able to see anybody. Okay, so thank you very much for the opportunity to be able to talk to you all today. I last spoke to a tree audience a solely tree audience in 2015 at a Harvard conference on the genetics of rainforest trees. So it's good to be back speaking to tree people. So we're going to, I've decided to do a double act today with Rowan, because although I've been working on Inga for about 20 years, Rowan has been doing some really nice work very recently with genomic analysis, and he's been doing that analysis himself using programs and procedures that I know nothing of, and I didn't really want to be trying to explain his results to you when he could explain them so much better. So a double act. Right, so what you're seeing here is a view through the Amazon rainforest, which you can hear, unfortunately, but not see, because that's the limitations of our technology at the minute. This is part of the Yasuni National Park in Ecuador, where Rowan was doing field work earlier this year. Was it last year?
This was my PhD field.
Ah, it's your PhD field work. But it's a beautiful place, but what I want you to be aware of is that all the trees you're looking at in this scene are all likely to be different species. The diversity is that high. It's completely unlike what you'd see in a temperate forest. So there are more tree species in 1 hectare of Amazonia than in all of Europe. It is extraordinarily diverse, and that diversity is on a really fine scale. So even within very small patches, you've got huge numbers of species. And this is one of the things that we want to be able to understand. How is this diversity created, and how is this diversity maintained? And to do that, we're working with Inga as a model group. So Inga has a number of advantages as a way in to try and to understand this diverse and very complex system of tropical tree diversity. So they're legume trees, they're a nitrogen fixer, and that's one of the things that makes them into something that's useful for people, which makes them more interesting to study and more interesting to get funding for as well. So they're used as a multi-purpose agroforestry tree. They provide shade trees for coffee and for cocoa and for tea, and some of the species actually have edible fruit as well. So one of them produces what's known as the ice cream bean. So there's something that's generally of use. But one of the things that makes them useful for understanding diversity is that there's a fair number of them. There's at least 300 species, and they diversified very quickly in the last 6 million years. So if we can start to try and understand how Inga has done this, then this will give us a foothold into trying to understand diversity in the Amazon altogether. And as I was saying, that diversity in the Amazon is on a very fine scale, and you can see this in Inga. So this is a wee map showing Barra Colorado Island, which I still can't pronounce properly, in Panama. And this is just over 15 square kilometres, about 6 square miles, so smaller than the centre of Edinburgh. And within this area, there are 15 different Inga species that are found. So this is, and there's lots of them there, and they're found growing side by side in individual plots. And one of the things that can be a bit tricky about Inga is they tend to look very similar. Here's some botanical illustrations, and to my eyes, to not a taxonomy person, they all look much of a muchness. There are experts in the field, however, such as Toby Pennington, Toby Pennington's dad, Terry Pennington, and Rowan, who have got experience with these things in the field and can tell the difference, but it's very tricky. They also, they behave in very similar ways. You can't tell a nice little story about Inga speciation through changes in pollinators or changes in seed distribution. Their flowers mostly look like this, like little bursts of stamen, typical mimosoid legumes. So how can we tell Ingas apart? We need DNA to do so, but there are creatures that can tell them apart, no problem, and those are the things that prey on them. So this is some work by from Ndara et al. From a couple of years ago, which looked at the various different, in this case, sawflies that were predating on Inga species across the Amazonian area. And we have here on the left a phylogeny of Inga species, and on the right a phylogeny of the sawflies that were found on them. And the sawflies, there's so many of them, and they're so pitiful little things, they don't really have species names. They've just been dealt with as operational taxonomic units here. But what I want you to notice is that there are very specific relationships between individual operational taxonomic units of sawfly and an Inga species. So the things that eat Ingas can tell them apart easily. And Part of this is because there's a really strong relationship between the herbivores and the ingers. There is a huge amount of selective pressure on the ingers from the herbivores. So a lot of this happens in the very first weeks that a leaf is starting to expand, when it's actually vulnerable to attack, when it's setting up its defense systems and starting to grow its photosynthetic capacity. That's the time that it's most nutritious and easiest to access for attacking herbivores. So this is a very strong selective pressure at one particular developmental time in the plant's lives. And this has led to the trees trying to counteract this by putting an awful lot of energy and resources into defending themselves against herbivores. So over 50% of the dry weight of young leaves of some species is anti-herbivore compounds, a range of different secondary metabolites. And it's really interesting to see how Inge have gone about doing this. So one way they've done it is by using overexpression of simple metabolites. By hugely overexpressing tyrosine, you can poison the herbivores that are eating you, because they just don't need that much tyrosine. It bungs up their systems and they die. And this is some work from Colley et al. From a couple of years ago, showing the percentage of dry weight of a leaf that's tyrosine in a number of Inge species, and you can see that some species are really specialised in this means of defence, and the other lineages are not using it at all. And they've done this by modifying the gene expression pattern of the biosynthetic pathways leading to tyrosine. This is another way in which they've gone about, some species have gone about defending themselves against herbivores. So there's trypsin inhibitors that are found across legumes, often in legume seeds, and what these do is they bind to trypsin, blocking the active site and preventing it being able to break down proteins. So the more the herbivores eat, the less goodness they can get out of what they eat. So they starve themselves the more they eat. And when we first did some transcriptome analysis of Inge species, I was really astonished to find that over 5% of the reads in a transcriptome made from an expanding leaf of Inge umbellifera coded for trypsin inhibitor. So the Inge umbellifera has gone great guns into using this particular way of starving the caterpillars that are attacking them. And when we look at this across a number of different species, so what we have here is a phylogeny of these trypsin inhibitors across five different species. So this is Senna, which is the out group, and then in red we have trypsin inhibitors from Inge sapindoides. In green we have trypsin inhibitors from Inge spectabilis, and in blue we have trypsin inhibitors from Inge umbellifera. And you can see that this particular gene family has duplicated and diversified across Inge, and that in Inge umbellifera we've seen really high expression of at least two of these gene copies, and the spectabilis is expressing at a high level from one of these as well. So the Inges are investing in this particular pathway, and they're varying in how much effort they're putting into it. But those are very sort of basic ways of defending yourself against insects. There's much more sophisticated ways in what they do with their chemistry. So this is work from many years from Tom Kersar and Lucy Colley, looking at the chemistry in Inge leaves. And they do produce the most extraordinary range of chemistry. So using mass spectrometry, no, I'm giving up on that one. You can just read it. There have been a number of studies that have looked at the diversity of compounds that have been produced by Inge leaves, and here's another name down the bottom here for something that I'm not going to try and pronounce. There's a particular compound that has only in the universe ever been seen in expanding young Inge leaf. So they play with their chemistry a lot and produce things you just don't see elsewhere. And this chemical diversity can even be seen across a single species. So here's an example of various different populations of Inge umbellifera from across the Amazon, and through mass spec analysis you can see they produce very different sorts of compounds. And this is exactly the sort of within species diversity that provides evolution with a lot of material to work with. So the idea of the relationship between herbivores and the plants that their host plants has been thought about a lot for many years, and the sort of classic work is from Ehrlich and Raven in 1964, which suggested there'd be an arms race where you would see small changes in the host plant matched by small changes in the herbivore, then more changes in the host plants, and more changes in the herbivore, and you would see a sort of a stepwise sort of race together as the host and the herbivore tried to get one over on each other in respect to their defences. And this was the accepted way that evolution would drive these relationships. So given the information we had in Inge, we were able to test this, and this is work from Forrester from a couple of years ago. From a really nice Inge phylogeny we were able to produce, and Dale Forrester brought together all the mass spec work that had been done on Inge over the years and did a thorough survey. And this is showing the diversity in chemical compounds across the Inge phylogeny. And there's an awful lot of chopping and changing. And when you compare general chemical similarity in compounds within species, between sister species, and between species that are either parapatric, found in adjacent habitats, or sympatric together in the same habitat, you can see there's actually more variation between plants that are in sympatry than you would imagine, and that sister species vary greatly. So the whole thing is a lot more labile than is predicted by Ehrlich and Raven. And one of the things we think that might be important in driving this lability and allowing the Inges to chop and change their chemistry and experiment with producing new chemicals is the secondary metabolite clusters. So we've looked at the genome sequences of a number of different Inges. This is data from Sapondoides and Spectabilis that we ran through Plantismash to look for clusters of genes that are involved in secondary metabolism, and we found a fair number of them. This seems to be one of the ways that the Inge arranged their secondary metabolite genes. For example, here's a cluster from Inge sapondoides, and this is on got 11 of the genes that 11 genes that modify saponins productions, all arranged in a big cluster there in one part of the genome. Along with a couple of genes that are involved in producing the saponin in the first place. So Inge sapondoides has got a number of these clusters actually across the genome. Has concentrated the genes that modify these pathways, allowing them to be inherited and recombined together. And we think this is one of the things that Inge have been using to generate new chemistry. So it's very unusual for novel chemistry to be produced by new genes that suddenly do something that's unexpected. What happens more frequently is that you see a gene duplication and then a slight change in the substrate or or what the enzyme is producing, or exactly which residues it's going to modify. And these subtle changes are how you get changes happen in secondary metabolites, or you get a combination of two things together, then allows a new type of protein to be, a new type of substrate to be able to be produced. So we're interested in looking at how these gene clusters and biosynthetic genes have evolved across Inge, and whether this has given them the capacity to be able to outrun the herbivores. And this is where I'm going to hand over to Rowan, because he can tell you in a lot more detail about a project that he's been working on for the past 2 years to understand how Inge are doing this.
Hello everybody. Yes, I'm Rowan. Thanks, Catherine. So, so with in mind of these this combinatorial nature by which chemical defenses evolve in Inge, the natural question is then, are these blocks of chemical defense genes being passed around between species, and this admixture, is that leading to higher variation in these traits, which can then spark this extraordinary diversification? And hybridization has been thought of something that can more usually erode diversity rather than generating it. But despite that, it's becoming ever more clear that hybridization is this key catalyst of some of the most amazing rapid evolutionary diversification events in our planet's history. This is because Hybridization brings together novel allelic combinations that would never happen usually by chance, basically bringing together allelic combinations from different parental species that are usually adapted to different ecological contexts. This means that in the descendant lineages, genetic variation can accumulate much faster than by mutation alone, and when coincident with ecological opportunity, this can spark this sort of rapid phenotypic evolution. A classic example in plants being Hawaiian silver swords, this sort of hybridization between a pair of California tarweed species colonizing the Hawaiian Islands, leading to this really diverse radiation event with lots of different ecological guilds. There's these arborescent species, rosette-forming species, cushion-forming species, climbers, sort of almost epiphytes. It's incredible. And it's really interesting because much of the extraordinary hyperdiversity of rainforest trees is due to exactly these sorts of recent species-rich evolutionary radiations. But despite that, the influence of hybridization on rainforest tree diversity, particularly in the Amazon, has been almost totally sort of overlooked in a sense, probably because of the sort of methodological difficulties. But there is novel evidence emerging that hybridization is playing some sort of a role in this, generating this extraordinary diversity. And so the title of the main question behind my postdoc is whether hybridization is a major driver of evolutionary radiation in Amazonian trees. And as Catherine mentioned, I'm using this amazing system INGA, which as a sort of typifies the extraordinarily rapid radiations that make up most of Amazonian tree diversity, with 300 species diversifying in the past 5 or 6 million years. And INGA are really ubiquitous. They're more or less everywhere in the tropical Americas. You walk through a forest, you'll see about 5 INGA species just on one little path. And in addition to that, as Catherine was mentioning, they're extraordinarily diverse in terms of their defences, which is really typical for rainforest trees because of the sort of constant onslaught of herbivory that they're exposed to. And it's to the point that herbivory really structures the extraordinary diversity of tropical trees we see in rainforests. As per the Jansen-Connell hypothesis, if you're a tree species and you become highly abundant, so too will the abundance of your herbivores increase, bringing you back down to sort of a low level of abundance. And this sort of penalization of high abundance is what leads to these extraordinarily species-rich but sort of low-density communities we see in tropical trees, particularly in rainforests. And of course, this constant herbivory drives this huge selective pressure for defense. As Catherine was mentioning, there's a lot of chemical diversity for defense in INGA, but as well as that, we know that defense chemistry is this key axis of divergence and most crucially, coexistence in INGA. The reason that these hugely diverse communities can accumulate, for example, up to 20 species of INGA can be found in a single hectare of the Ecuadorian Amazon, is because each of these species has a different chemical strategy for defending themselves against herbivory. This sort of, again, back to this density-dependent pattern of Jansen and Connell, novel defenses mean survival. And this makes sense because if you think about it, if you're an INGA and you disperse into a region with lots of your conspecifics who have the same defenses as you, you're all going to get eaten. However, if you have a new chemical profile, for example, if you're the introgressant descendant of a hybridization event with a new combination of defense chemicals, and you disperse into the same area, the local herbivores won't be able to digest your defense compounds, whereas your friends are still going to get eaten. And with this in mind, I'm sort of really interested in whether admixture of these loci that underlie these diverse defenses, when coincident with ecological opportunity, in this case escape from herbivory, has helped to catalyze the rapid diversification of INGA into all of these extraordinarily diverse species. Okay, so how can we test this? Well, we can, I guess, basically take 2 broad approaches. We're basically looking for, first off, introgression, of course, of defense loci. So in other words, for those uninitiated, introgression is basically the more or less permanent transfer of genetic material between lineages through recurrent hybridization and backcrossing. So obviously we need to look for hybridization in these loci. And then we can look for whether selection is maintaining these loci because of their adaptive effect. And that means we can ask two questions. First of all, of course, did introgression occur in Inga at all? Is it a good line of reasoning to go down? And secondly, are these chemical defense genes actually showing evidence of sort of preferential transfer, in the sense that after they're transferred via hybridization, are they being sort of selected for and maintained in the descendant lineages? For the first portion, I am addressing this with a broad phylogenomic data set with hundreds of Inga species, sequenced at thousands of phylogenomically informative loci. And then the second question, looking at introgression of these and selection on these defense gene blocks, using a large whole genome resequencing data set, sequenced at more or less population level for many Inga species. So let's address the first question. To do that, I've inferred an astral phylogeny, so basically a species tree, from 1,320 target capture loci for 190 Inga species. And that target capture data set also includes loci that underlie chemical defense or are associated with chemical defense in Inga. Then we can assess the incongruence between different gene trees. In other words, we can see whether the different gene trees tell the same phylogenetic story, in a sense, and whether they conflict and tell different stories using quartet concordance scores. And then we can test whether any incongruence, any conflict that we find, is better explained by introgression or incomplete lineage sorting, using tools such as d-statistics, which explicitly test for introgression. And once again, just to explain, incomplete lineage sorting is just the random inheritance of different sets of alleles. That's really very common in rapid diversification events, which means that these alleles don't sort nicely into their respective lineages, meaning that there's just this baseline level of conflicting gene trees. But the difference is that these gene trees are more or less at similar frequencies. And in this case, incomplete lineage sorting more or less acts as our null hypothesis against which we can test for introgression. So here's the resulting tree, built from all of these phylogenomic loci. And here we can see in the early divergences in the Inga tree, there's lots of green nodes, which indicate high quartet concordance scores. In other words, all of the gene trees are telling more or less the same story at these loci. There's minimal conflict. They all resolve more or less similar relationships. However, what we're interested in is the cases where there's one conflicting topology that is much more frequent, which is this very sort of a strong signal of introgression. And this is indicated by red nodes with negative quartet concordance scores. As we can see in the later divergences in Inga, and particularly towards the tips, these sorts of very negative quartet concordance scores are fairly common. But this QC method is not sort of an explicit test of incomplete lineage sorting versus introgression, despite the fact that this one conflicting topology is fairly strong evidence of introgression. So we can do one better than that. We can test for introgression between every pair of species in our tree using d-statistics. Some of you may know them as Abba-Baba statistics. In short, this is basically just asking whether there's evidence of introgression between every possible pair of species, here plotted as a phylogenetically ordered pairwise heat map. As indicated by the legend, if the blocks with more red and that are more saturated in colour indicate a more significant D score, in other words, more evidence for introgression. Here, most crucially, using a relatively new method, I'm accounting for differences in substitution rate, because these differences in substitution rate can unfortunately cause phylogenetic incongruence, which really mimics the signal of introgression. But here we're trying to account for it with this Abba clustering method. And to make it even more stringent, I've corrected each of the p-values, which are indicated by the color of the block for multiple testing on each of these jackknifed replicates. So, almost as sort of stringent as we can get. And despite that, there's still evidence for this sort of hum of introgression across Inga. There seems to be a lot of evidence for it. So, to answer the first question, did introgression actually occur in Inga? Well, it seems like introgression is fairly widespread in Inga, at least nominally based on this phylogenetic evidence. But does this actually lead to, do these hybridization events and this introgression actually lead to cases where individuals have a selective advantage, for example, through passage of chemical defense loci? Well, that's where question 2 comes in, whether these chemical defense genes are sort of preferentially transferred and maintained by selection. So to address this, I resequenced more than 530 whole genomes from 40 Inga species. These were sampled from the same replicate communities across Amazonia at population level. And in addition to that, I was lucky enough to be able to work with the Sanger Institute to generate 3 annotated Inga reference genomes. In addition to that, for these same communities, we also know the defense chemistries of these different Inga species and which insects eat which Inga species. And that's through decades of fantastic work by our amazing collaborators that Catherine was mentioning earlier on in the talk, Tom Kersar, Lissy Coley, Maria Jose Endara, and Dale Forrester. So, how do we actually find out whether there is preferential introgression and maintenance of these defense loci by selection? First, we can see whether defense loci themselves are actually passed between species in sort of contiguous linkage blocks or sort of cassettes of genes that go towards the same metabolic function. So first, we can use an even more stringent method for testing for genome-wide introgression, which is really nice, but because it accounts for correlated scores in terms of introgression among related taxa. In other words, if there's an introgression event at the base of a small clade of taxa, all of those descendant taxa will inherit that introgressed variation. However, this method, F-branch, accounts for that. Okay, once we figure out whether our genomes show genome-wide evidence of introgression, we can estimate more or less per gene or per window levels of introgression using a method that's much more sort of without the biases of D statistics when you apply them to small genomic windows. These D statistics, Abba-Baba statistics, are very biased when you apply them to small windows, so we use this method FDM to estimate how much each window of the genome has introgressed. And then just to visualize it for you, I've also done some per window PCA, which basically just groups individuals based on their genetic similarity in nice windows across the genome. And then to test whether those blocks are maintained by selection following introgression, we can use these classical methods like looking at divergence between windows of the genome, between species pairs that are introgressing, looking for selective sweeps with big blocks of highly homozygous fixed loci shared between species, and really nice tools like Volcano Finder, which look for these homozygous sweeps with these sort of volcanic peaks either side of heterozygosity indicating adaptive introgression. But these are still in process, so I won't be presenting those to you today. I was just highlighting that that is also being taken into consideration. So here's some results that are really hot off the press. Okay, so which species in our genomes, in our whole genomes, have the strongest signatures of introgression? Again, using F-branch, this extremely stringent method that accounts for introgression on related branches. And here, just to contextualize this is really, with all of these huge data sets, whole genome data sets, it really helps us to pinpoint which individuals or which species are introgressing. And so you can see, again, this sort of hum of introgression across Inga, but it's really concentrated in these short-lived, chemically diverse species in the red hair clade, what we call the red hair clade, these sort of live fast, die young denizens of trefoil gaps in disturbed areas in the Amazon, which are subject to incredible amounts of herbivory. Makes sense. So, okay, there is genome-wide evidence of introgression. So which regions of the genome have the strongest signatures of introgression? And I'll take you through a particularly lovely example. So first of all, I did 19 FDM analyses for 19 separate species trios, and these species trios were selected based on which species showed highest evidence of introgression with F-branch. I then took the 99.95th percentile outliers, so these sort of really, really outlying high introgression regions of the genome, and about 25% of those millions of FDM windows, about 25% of these outliers were in or very close to genes. Now, here's that particularly lovely example. I can just pull one out, which makes most sense, in a sense. So here we have introgression, this FDM score, plotted on the x-axis, on the y-axis, and then that's plotted across the genome in windows. And so a highly negative or highly positive FDM score just indicates high evidence of introgression in that window. Positive or negative just indicates which species pair is involved here. So in other words, the 0 point means no introgression, highly negative or highly positive means lots of introgression. Here's on the left-hand side, Inga capitata and Inga acrayana, this species pair from the populations in French Guiana. On the right-hand side is the same species, Inga capitata, with a different species, Inga tenue calyx, in Peru. And if we zoom in here on the PTPRR gene on chromosome 1, which codes for tyrosine phosphatase, as Catherine was mentioning, tyrosine is an important defense chemical in Inga. We see that there are these hugely outlying FDM values indicating huge levels of introgression in these blocks of the genome, specifically in this gene. What's particularly interesting from a sort of biological and ecological standpoint is that there's many ecotypes, about 4 or 5 ecotypes of Inga capitata. The only one that uses tyrosine is the LA12 ecotype. And this ecotype is found in French Guiana and Peru. And this is the only region in which we found outlying levels of introgression in this gene. So it sort of hints that there is some evidence of something biological going on, something interesting is happening. Similarly, if between these two species, if we plot their similarity across the genome on chromosome 1, most of chromosome 1, they're fairly divergent from one another. However, if we zoom in on this region at about 22 megabases, we see that their traces almost totally overlap. In other words, they have more or less exactly the same copy of this region of the genome. And more specifically, this is where the PTPRR gene is located, this gene that codes for tyrosine phosphatase. It's not just that one case. There's actually multiple cases of sort of replicated introgression of the same genes in different communities or even between different species pairs. And here I'll just plot for chromosome 7. I've tallied up all of the FDM outliers, so the introgression outliers across all runs and all species pairs, and then plotted them on the y-axis, and then on the x-axis their position on the genome. And we see on chromosome 7 there's this block of the genome that has way more FDM outliers across all of the runs than you might expect. If we zoom out across all 13 chromosome pairs in Inga, something very interesting is happening on chromosome 7. It's just this huge outlier for introgression, it seems, across all of the species pairs. This is still even the case when we account for the density of repeats, which might cause, for example, mapping errors, the density of genes, which may then influence the density of FDM windows because it's based on numbers of informative SNPs. Even accounting for those, these outliers are still really focused in this 20 MB block of chromosome 7. As to why, I'm just about to get into that, so stay tuned. I'll hopefully be able to let you folks know soon. So yeah, to summarize question 2, are chemical defense genes actually being transferred between Inga species? Well, it seems like yes, there's this emerging evidence of defense gene transfer between species, so watch this space. Hopefully we're going to be making some interesting discoveries very soon. And so overall, just to summarize, this project has really highlighted the power of not only the genomic revolution, but also collaboration with these amazing scientists across Latin America and beyond for opening this sort of novel research frontier, which was previously more or less impossible in the Amazon. And for that reason, this is among the most extensive genomic studies of introgression and speciation in the Amazon yet. We were fortunate to be able to generate a massive data set of whole genomes, just through collaboration. It's really fantastic to be a part of it. And I think this is really crucial. These big, I guess, blue sky projects are really crucial because, in my view, to understand speciation, you really have to understand the most species-rich regions on Earth, places like the Amazon. So yeah, thank you so much to all of the funders and amazing PIs, co-Is, the collectors of all of the silica material, folks that help with fieldwork and everything. And yeah, just to say, the first portion of this talk is on bioRxiv now, the phylogenomic portion. Cool.
Perfect.
Can stop
Yeah.
sharing.
Thank you very much.
Thanks. That was perfect. Yeah. Thank you very much, both of you, for speaking. Really interesting stuff. And yeah, just if you speak long enough, do you get a scarf, Rowan?
It's socks. Socks for Christmas.
No, that was very good. So can I open it up now, and we'll just allow anybody from the audience who would like to ask a question to please go ahead. We'll keep an eye on the chat as well, just to see if there's anything, if you want to type rather than speaking, please do so. Miriam, please go ahead.
Yes, hello. Thank you very much, Catherine and Rowan, for a very great talk. I was wondering, you spoke of a single ecotype that occurred in two very different regions. We're not used to hear about ecotypes in that way. Usually we think of them as a single species occurring, for example, in a small region, and we have different habitats that are very close by, where one is metal tolerant, the other one no, something like that. But you're talking here of a single ecotype that occurs across thousands of kilometers. How is the genetic diversity in this species structured, and how is it possible that an ecotype is in such different places? Are these really genetically closer, or are you defining the ecotype on a specific trait? I was just with all these questions, so I ask you.
They were referred to as morphotypes for the longest time, these slightly different versions of Inga capitata that kept turning up in different places. And it was one of the questions that we really wanted to address with our phylogeny is, are these genuinely, different groups, or is it just, a bit of sort of population level variation that we're picking up? But it does turn out to be different groups.
It does, yes. They group phylogenetically. There is some degree of geographical structure, but I guess the key thing that keeps them together is that it's the same morphology, so you can identify them in the field. And in addition, I guess most importantly, they share the same chemical defense strategy, in this case being tyrosine, which is atypical for Inga capitata. But yeah, I guess in terms of structure, they are each other's closest relatives, which is quite interesting. And I think it speaks to the extraordinary ability of Inga species to disperse. They're dispersed by animals, and they have more or less free run of the entirety of the Amazon. Any one community in the Amazon is composed of a species pool that basically is effectively the whole Amazon, which is, I think, quite interesting.
So you could make a case for making them species, if you wanted to mess with the taxonomy.
Yes.
Yeah, it's not typical for legumes to be known as very effective dispersers, so that's very interesting. Thank you.
Great, thanks, Miriam. Anyone else? While we're waiting for everyone to think, could I just maybe ask, do you have a sense of where the boundaries of hybridization lie across the phylogeny? Is it and in the particular pairs you were looking at, is it free hybridization between the two species, or do you get asymmetric gene flow one where your hybridization one into the other?
So that's why that's a really good question. So that's something I'm looking at the moment between a set of really closely related species pairs. I'm using a method called Gimbal, which does demographic inference of patterns of effective population size and migration across the genome. It was developed by Konrad Loser here in Edinburgh. And it basically fits a model of strict isolation or divergence with gene flow across the genome. And what that means is that we can pick out barriers to gene flow across the genome, and in some cases, hopefully in the new version, directions of gene flow. So hopefully I can answer that question more effectively between at least a few species pairs quite soon, if that makes sense.
No, it does, absolutely. Any others? Ivan, please go ahead.
Hey, hello. Thank you very much. Very nice talk. Didn't you manage to date the introgression events? Is there a way that you can do it?
It's extremely difficult for various reasons, mostly, I guess, incomplete lineage sorting and the very recent radiation, meaning that I guess the error bars on any phylogenetic dating would be massive, particularly because we don't have any fossils in Inga. But I think the way that I could do it, and I might try, is by using demographic inference. Then we can at least get an idea in terms of how many generations ago we had introgression among some of these species pairs, and then we can very roughly convert that into years.
At least understanding whether it is very recent or very ancient or something like that.
Yeah.
Thanks.
That's part of my fellowship plan, so hopefully that gets funded.
I was just wondering, is there going to be a gap? Is there a fair gap between what you can do with demographic inference and what you can do with phylogenetics? There's this sort of space in the middle where it's got to be millions of years for phylogeny and there's tens of thousands for demographic inference.
There is, and then even with demographic inference, it's more difficult with Inga because they have huge effective population sizes, which means that the divergence time is probably less than 0.4 times any, which is sort of the key, I don't know, threshold for demographic inference, for coalescence events to be able to tell you what's going on. So how effective demographic inference will be in Inga, we'll see.
We will push the boundaries of the possibilities of demographic inference.
I think so. Yeah.
Question from Mo.
Yeah. I think there was just one comment in the chat there. The introgression dating would be difficult because there should also be multiple phases of introgression throughout the divergence times. Point.
Yeah.
Sorry. Yes, go on.
I see there's a comment about the syngamion, which is arguably my favorite model of introgression in Inga. These co-occurring, interfertile, extremely closely related species that, reduce allele effects by increasing genetic diversity and through introgression with each other, like oaks, the classic system. But we'll see. Thank you, Mong, for the comment.
Thank you. Yes. So Nicholas, go ahead.
Hi, thank you very much for a great talk. I was wondering, how do you characterize the community of insects that eat the Ingas?
So the insects were collected and characterized by barcoding. We realized that there was no way they were going to be able to sort of taxonomically get everything, so they were roughly sorted into groups and then used Cox-1 barcoding.
Okay, so they collecting them on the plants.
Oh yeah, it was an awful lot of work. It's very impressive.
Okay.
So the plants, they on individual plants, they collected the insects. They took leaves for doing mass spec analysis, and they sent leaves to us for doing transcriptional analysis. So we got a really nice, rich data set.
Wow.
That's why I mentioned these amazing collaborators, Lissy Coley and Maria Jose Endara. Their work is amazing. And just to sort of contextualize that insect diversity, I've seen an estimate that says there's an estimated 100,000 insect species in a hectare of the Ecuadorian Amazon, which is more than all of North America, which is like unbelievable. I don't know if that's true.
Yeah. Okay, well, thank you so much. Excellent work.
Thank you.
Thank you. So Catherine, you're next, I think.
Hey, thanks for a nice talk. I'm happening to also collaborate with Majo Endara in the Ecuadorian Chocó region, and just out of interest, for which of the Inga species did you then generate genomes, and for what reasons how did you select them of all those many that you had available?
So first of all, we generated with Sanger Institute chromosomally contiguous annotated reference genomes for basically a few of the species we had in Edinburgh. They were chosen, first of all, 2 of them were chosen because they were diploids, and it turned out one had a massive inversion, which is very interesting. And then we chose the 3rd species because it was apparently autotetraploid, and it was indeed. So that's Inga laurina is the autotetraploid Inga Lyocalycina and Inga oestediana, and those genome notes, those genomes are available now. We just released the
Cool.
genome
Yeah.
notes. And for the whole genome resequencing, that was based on 3 criteria. First was based on the phylogenomic analyses, which species showed evidence of introgression. Based on my initial rough estimates of ploidy based on site frequency spectra from the phylogenomic data, I wanted to see if any Inga were polyploids, so I targeted those, but not very many of them are, which is helpful. And then I also targeted species for which I would like to ask these demographic inference questions.
And then we have the Todd Michael genomes
Yes.
as well. So way back before Rowan joined the project, Todd Michael offered to do a couple of genomes for us of material that he had available to him through the Smithsonian. And that was where we got draft genomes of Sapondoides and Splendiferous. Not Splendiferous. Spectabilis. Yeah, they're draft genomes, but they're still very useful for comparison.
Thank you.
Great, thank you. There's a couple of interesting questions in the chat actually related, but and it relates to something I was going to ask myself actually. Is that what do you suppose? So this from Jeff Garnas, and Kit had a similar question. But what do you suppose maintains species integrity in Inga? So if you've got this sort of soup of hybridization going on, what is it that allows something to maintain itself as an identifiable species? Now, does that maybe relate to the question of, are there evolutionary strategies, I guess, of hybridization avoidance or being a hybridizer that might actually benefit you in that sort of context?
Yeah, it's a really, really interesting question, and one I've spent a lot of time lying awake at night thinking about. So I guess in the most broad sense, I would say that it might be that it's something like genomic incompatibilities, or it might be that it's something like, or even it could be between diverging sister species. It could be this ecological divergent selection like we found from Catherine and Dale's work at the level of sister species divergence, with, say, defense loci acting as barriers to gene flow. And then later on, despite the fact that at this local stage, hybridization is prevented between chemical defense genes that are so crucial to lead to divergence, then later on, after they accumulate more divergence across most of the genome, in some rare cases there might be hybridization which is useful, but maybe most of it is just across other parts of the genome. So that's where it's going to be super interesting to get more into this FDM and selection stuff.
And to see which bits of what compatibilities there are between the different alleles and different loci.
Yeah.
Another thing that was discussed quite a bit amongst the AIM group is edaphic factors and the sort of soil types across the Amazon, and more general sort of abiotic. So abiotic factors, flooding and this kind of stuff. And there we just wouldn't know where to get started with the candidate genes. But clearly there's not a big signal like there is with the defense loci, otherwise it would have come up. Yeah. We would see it. So there's more subtle effects going on there.
It's interesting because almost all Inga species have very, very similar flowering strategies. Their phenologies overlap, they share pollinators. So like there's an ecological reason for why they would locally hybridize. But I guess one of the questions I hope to address in a potential future fellowship would be whether these structural variants, like we found in this reference genome, is a massive structural variant in one of the species, whether these structural variants prevent recombination and prevent gene flow, and maybe it's structural variation in the genome that keeps these species apart. Hopefully we can address that soon.
And to look at the genetic structure in the seeds that are produced versus saplings that exist versus adult trees, and to see where the sort of environmental filtering is kicking into this as well.
Like the Lyocalycina system. That would be awesome.
That actually, so that relates to it. I was going to ask a simple question about where selection's actually acting. So is the herbivory strong enough on an adult tree that you just see, is the knock-on consequence just lower productivity, reproductive output from an individual? And does that differ between what might happen to a seedling? And in consequence, do you get different sort of chemical defenses in seedlings versus adults?
Yeah, you do. So the things that we've been looking at are all on the expanding leaves of understory things. No one's been looking at mature trees, and there could be a very different picture going on in mature trees. But, sorry. But the pressure is so strong on that sort of, whether a sapling actually ever gets to make it to a mature tree, that's where all the effort has been focused.
Yeah, so Dale Forrester and Catherine and Lissy and all these amazing authors have a lovely paper in Science that shows that herbivores cause something like 90% mortality in seedlings. It's like at the seedling stage, it's where it's really this crucial, I guess, bottleneck filtering step.
But actually, whether how many seeds and what the reproductive capacity is of a mature tree and how that's affected by herbivores, I don't think people have looked at that.
No, I guess not.
Certainly, they haven't looked at it in the Inga that I've read about. So yeah, it would be really nice. Particularly since there's a lot of investment in defence in seeds in Inga as well, and filling them full of interesting compounds. So
great, no, really interesting. Sorry, another hand, Christian, yes.
Thank you very much for that very nice talk. I wanted to ask you, we're concentrating now on divergent selection, but have you also looked at signals of balancing selection? When I think about defense mechanisms or immune system, I could imagine that heterozygotes have actually an advantage. And did hybridization could actually favor this balancing selection? Did you look at that?
Oh, we're back.
Hi, Christian.
Yes.
We lost you for a moment.
It's amazing. When I wanted to ask something, I got kicked out.
Maybe
I wanted to ask if you have looked at signals of balancing selection, because when I think about defense mechanism or immune systems, I immediately think about if you're heterozygote, if you have broad mechanisms, you might probably be better against this herbivory. And this would speak for hybridization, actually pushing a balancing selection, so pushing the heterozygous state. So have you also looked at balancing selection and only divergent one?
I haven't looked at balancing selection, but I've had both a few reviewer comments and just my own personal thoughts. I want to sort of take into account how density dependence interacts with these effects, because we have these super strong density dependent patterns in just herbivory and all that. I guess it's kind of similar, but the opposite to the heliconius butterfly system, where there's density dependence in terms of if everybody has the same mimicry gene, then it's advantageous, but the opposite is true for Inga. Rarity is favored, which I guess would drive up balancing selection. You want a higher diversity of lots of different alleles, right? But
also another reason for looking at copy number, because it looks like there's a lot of tandem gene duplications in these loci, and that'll be a way of hardwiring your allelic diversity. Yeah. I mean that SLC locus where you've got 11 copies of the gene.
Yeah.
You've got possible 22 different alleles there. Lots to play with.
Awesome.
Yeah.
Great. So any other questions? I think if not, we're probably coming to quite a nice natural endpoint at the hour. So I think maybe I know there are lots of people in the room with interests in near-tropical tree genomics, so I'm sure that's been really fascinating for lots of people.
Yeah, people have questions and want to email us, we'd be really happy to apply. It's lovely to have an opportunity to think and chat about this.
Yes.
Okay, great. I'm sure there will be follow-ups. All right, well, it just remains for me, I think, to say thanks very much again for the talk. Really interesting stuff, and we look forward to what has yet to come. And just to remind everybody that the next seminar will be in 2 weeks' time from Jens Christian Svenning, and I hope to see you there. So thanks everybody for coming along.
Thanks
Right,
very
just
much.
Thank you.
Cheerio.
Thanks so much.
Bye.